Good afternoon, members of the research committee, clinical colleagues, and faculty peers. Thank you for inviting me today to discuss my research. As a researcher, working at the intersection of biophysics and clinical practice, my work addresses a critical bottleneck in cardiology. How we translate low structural anomalies into predictable, actionable diagnostic indicators. Today, I will demonstrate how my research resolves this challenge by building a purely human-centric translational engine. To begin, my presentation is titled Clinical Arrhythmology, Bridging Anatomical Origins and Electrocardiographic Waveforms. I have no financial relationships or commercial conflicts of interest to declare. By focusing our specialty on human electrophysiology, clinical mapping, and human parameter modeling, we eliminate the traditional translational gap from the outset. Mapping arrhythmias, origins in heart anatomy. Moving to slide 2, we map these arrhythmias across human structural topography. For the clinical directors in the room, this hierarchy detects your daily management workflow. Shifting from a localized rate control focus in the upper atrial syncytial down to an acute survival focus when pathology strikes the ventricular myocardium. For the basic scientists on the committee, we treat this structural map as a multi-scale biophysical engineering challenge. We take high-density 3D coordinates from active human ablation suits to anchor our computational tissue masses, mapping exactly how microscopic ion channel kinetics in this specific quadrant scale up to detect whole organ surface ECG behavior. Four clinical categories of irregular heart rhythms, arrhythmias, are premature beat, supraventricular arrhythmias, ventricular arrhythmias, and bradyarrhythmias. This slide seamlessly integrates the four visual cards with the acute clinical emergency framework. This structural mapping brings us directly to our primary clinical challenge. While these cards classify the standard clinical landscape, we zero in on the high-risk pathology highlighted in box 3. Ventricular arrhythmias and its direct catastrophic progression to the endpoint on the right, sudden cardiac arrest via hemodynamic collapse. For our clinicians, you know how rapidly stable ventricular tachycardia can break down into chaotic ventricular fibrillation. For our scientists, our research models this exact moment of shock as a multi-scale biophysical failure. We study the precise boundary kinetics where coordinated myocardia squeezing breaks down into chaotic, non-coordinated reentrant loops. By understanding this mechanical chain reaction, we shift our clinical approach from reactive intervention to proactive predictive interception. Research engine, the human in silico data pipeline. This brings me to the core machinery of my research program, the human in silico data pipeline. To achieve a truly predictive framework, we deploy a strict three-step loop using human-derived data and computer simulations. In step 1, we ingest high-density 3D electroanatomical mapping coordinates directly from human clinical ablation suites, leveraging active international patient datasets from different countries. In step 2, this human structural geometry parameterize our multi-scale computer models running on high-performance computing clusters. We model cellular mechanics exclusively using human ventricular cell kinetics, specifically utilizing the O'Hara-Rudy action potential equations. Finally, in step 3, our model solves the mathematical forward problem to calculate digital 12-lag waveforms. This allows us to accurately map and predict the exact anatomical locations of reentrant tissue breakdown, providing a purely human-centric diagnostic output that can directly integrate with existing hospital software. Diagnostic and therapeutic pathways, where research intervenes. Coordinate axis, dual waveform tracers. We do not stop at calculating abstract tissue map. As a principal investigator or a researcher, my priority is ensuring our data models translate directly into procedural and engineering assets for active clinicians, deploying through 2 specific entry channels shown here on slide 6. Under ablation translation, our predictive spatial tracking algorithms convert standard surface voltage vectors into precise 3D spatial coordinates within the active part anatomy. This moves beyond traditional qualitative interpretation, isolating structural precursors and guiding cathedrals directly to focal reentrant triggers. Simultaneously, under device synergy, we integrate our advanced tissue waveform kinetics into the firmware layer of implantable devices. By providing biophysically detailed human datasets, we improve the adaptive pacing and sensing algorithms utilized in implantable cardioverter defibrillators (ICDs), enabling faster, more precise recognition of ventricular fibrillation, while minimizing the risk of inappropriate therapeutic shocks. To conclude, I want to step away from the specific data models and outline my strategic vision. During the initial phase, we will establish our high-performance computational cluster and standardize our parameters using our unique human data cohorts from countries such as the United States, United Kingdom, and China, ensuring our simulations capture vital structural variations. Ultimately, the true value of our research lies in clinical utility. We will form an active technological bridge with your hospital's clinical electrophysiology division, taking our predictive algorithms straight into the mapping suites to cross-validate our coordinates against real patients' outcomes. By bypassing animal models entirely, this independent research program provides a direct pathway to bring advanced digital diagnostics directly to your institution. Thank you for your time, and I'm now happy to open the floor and take your questions.